It is Thursday, July 9th, and you're watching the Biotech Voyager. It's the show that covers what's next in biotech, covering the early stage signals that are going to be a big deal in a couple years. Thank you so much for joining us. If you have any thoughts throughout the show, feel free to drop a comment down below. I'm joined, as always, by Jeff, the expert of early stage. Really excited
How
for
do you
the show
do?
today. I'm doing well. Great mug. Love that. I need a mug like that.
It should just say Jeff's blood on it.
Jeff.
All that it is. There's no difference between black coffee and Jeff's blood at this point.
Jeff has a blood coffee percentage of 5%.
One. It's one.
It's one.
Just 100%.
Phenomenal. All right, excellent. Well, ladies and gentlemen, we have a pretty great show lined up today. A lot of really interesting signals to cover this week, and then later on the show, we're going to be having a special guest, Reagan Jarvis, the CEO of Anaka. Anaka being a really cool company doing some pretty cool stuff in the TCRT space. So,
Yeah, they dosed their first patient on June 11th.
Excellent. That deserves an applause, wouldn't you say, Jeff? Absolutely.
We will give them one later too, but go ahead. There you go.
Congratulations. Good job. All right, excellent. So, folks, let's just jump off with the early stage leaderboard review, and then we'll get into some of the key stories today. The first one being the Sino shock with AstraZeneca, Jeff. Not a typical early stage signal, but I think actually has a lot of bearing for the companies that we'll be covering it regularly here on the show.
Yeah, so you mentioned this to me this morning, and I was like, who is what? Because I'm just like in early stage. So you're going to have to teach me what is going on here.
For sure.
I have no idea what's happening ever with most things in my life.
No worries. Cool. But yeah, just right here off the bat, Oligos Therapeutics has jumped to the absolute top of the leaderboard. Jeff, what's going on with them?
Yeah, well, so here's the dealio. Here's the thing. We mentioned them on Tuesday. The board hasn't changed much since Tuesday because the Biotech Voyager operator, aka the expert of early stage, aka Jeff, has been busy and hasn't done his due diligence on maintaining signals. So most of the signals that have come in have kind of landed companies in like the 20 to 30 range. So not much of this has changed. However, we can go and look at those signals if you'd like. Let's pop open those early stage or these recent signals. So there's one that I wanted to maybe pop open, and it was this. This looked kind of interesting. AI-driven small molecule discovery initiative in Tel Aviv. Let's take a look at Evogene. Do you know Evogene at all or no?
I do not.
Me neither. Let's learn about them.
When I was looking through the signals for today, that was one that I passed over sadly.
Yeah, it's relatively low. Well, the actual signal here is they launched an AI-driven small molecule discovery initiative with Tel Aviv University out in Israel. Let's see. Computational biology company using ChemPass AI platform, a generative AI engine built on 38 billion molecule foundation models to design novel small molecules for pharmaceutical and agricultural applications. That's cool. Its lead programs target oncology, including aggressive breast cancer and non-small cell lung cancer, cisplatin resistance, immunology, irritable bowel disease via dysregulated neutrophil activity. That's cool. And crop protection, novel herbicides and fungicides via subsidiary Ag Plenus. Ofer Haviv has served as president and CEO since late 2004. Ben, this is an interesting use. I mean, we're in like the therapeutic space, right? But I always forget that biotechnology extends well beyond like technically speaking, like human health or human therapeutics. I was working with a company who was a biotech company, and their big thing was fertilizer. And it was biotech. And I was like, fertilizer's not biotech. But then I was like, wait a minute. It is absolutely biotech.
It totally is.
Yeah.
So this is actually really interesting because back when I did my master's in biotech at the University of Guelph, the University of Guelph.
I'm Ben and I have a master's degree. I cannot talk at all. Every third word is during grad school. So for me.
But no, the biotech program, like the University of Guelph is a big agriculture focused university. Mm-hmm. And so one of the major pillars of the different ways that you could use that biotech master's was in the ag field. There's so much cool stuff that's going on there.
Okay, perfect. What?
Yeah,
what's cool?
So for example, well, this is now dated, right? Because that was 6 years ago. I'm showing my age. But
I'm 30, so old. I'm showing my age. Anyways, continue.
But which is one of the really cool things that was happening at that point is they actually found out that when it came to like greenhouse production of a lot of these different plants, using basically like sparkling water, like CO2 infused water, and spraying that on the plants dramatically increased a bunch of different factors for like productivity of plant growth and stuff like that. There's so many just really cool applications of
I feel like that would be like a lowest hanging fruit, like because plants need water and they need carbon dioxide.
Yeah.
So you think like let's just take them both and put them together and put it on the plants.
Turns out plants like bubbly too.
Revolution. Okay.
Turns out the plants also like superior.
I tried to put a little bit of alcohol in mine and call it a day.
But all that being said, yeah, Evogene is using this small molecule AI platform for not only therapeutic development but also for crop production.
Interesting.
Super nifty.
Yeah. See, whenever I think of agriculture in biotech or like small molecules, I think of pesticides. I would never think of things that like, I don't know, help your carrots get bigger.
Right.
But I guess it would matter a lot, right? What else we got? Okay, so Evogene, perfect. Sounds okay. Vaccinax.
I have them later, so we can get to them a little bit later. Yeah.
Okay, perfect.
But if we just jump up next here to the Sinoshock, I think that might be interesting for you, Jeff.
You can take the board down if you need to.
Cool.
Yep.
Excellent. All right. Yeah, let's jump over to this one. Really interesting story here. So again, Jeff, I know typically Phase 3 is a graduation event for the companies that we cover on the Biotech Voyager so that we can focus on those early stage signals. I thought this would be an interesting exception to the rule for a couple different reasons. First of all, Sino Biopharmaceuticals signed a $2.1 billion with a B dollar deal with AstraZeneca to acquire their Phase 3 asset that's going after COPD. But here's the wild part. You ready for this?
No.
This entire program, currently in Phase 3, initiated its Phase 1 clinical trial in late 2022 in China.
Oh, 2022?
2022.
So 2023, 2024, 2025, half 2026 is like Phase 1, 2, and then what, half of Phase 3?
Yeah. And then got acquired by AstraZeneca for $2.1 billion. That's
I'm wondering.
Wild. Like, dude, like we need to just like stop for a second, right? Previously, the playbook for this kind of thing, right, is typically like 10 to 15 years in order for you to kind of get to Phase 3 and start doing some real work. They are at Phase 3 and bought by AstraZeneca for $2.1 billion in 3 years.
I would say 10 to 15 years is probably like discovery to approval, but
sure.
I hear you. It's probably cut the timeline by like maybe 30 to 40% faster.
That's just wild to me.
Yeah.
And I think in a lot of ways, just kind of, again, really turns up the heat for the competitiveness of these different regulatory agencies. I know this is something we've kind of talked about on the show in the past, but I actually do see this kind of thing primarily going through China and then a global pharma licensing it or acquiring this asset for $2.1 billion. It puts some interesting competitive pressure on agencies like the FDA because,
for sure.
Right.
Yeah.
What it means is, you've kind of got complacent as the world leader, but now significant amount of capital are going into these different therapeutic assets getting developed a lot faster in other parts of the world. You'd best stay on your toes in order to maintain that competitive edge.
Yes. Yes. One of the things that I'm wondering though, like what exactly is this thing? Because certain therapeutics take longer to develop than other ones.
It is a small molecule.
Okay. That's the first thing. Number two, there definitely is obvious advantages. Again, we get into like, I don't know enough about the actual differences between clinical trial design requirements in China versus the US and other areas. But it's one of those things where like, this is coming from a place of mild ignorance. So grain of salt.
That's all of us, Jeff.
Grain of salt. Well, okay, then significant ignorance, I guess.
That's all of us.
It seems like this has to begin with a presupposition that the FDA just doesn't know what it's doing and has been making mistakes for no reason for like decades. I tend to think that the FDA knows what it's doing and we have certain safety standards put in place, and to accelerate those things because it just takes too long to do this might be a little bit naive.
I generally agree with that sentiment. However, I think the other thing you just kind of have to consider, right, is the bloat of bureaucracy, and kind of being the top dog for a long time kind of induces additional workflows and like inefficiencies that may be well intentioned, but really kind of line up to death by 1,000 cuts over an extended period of time. So again, I do agree, right? Like the FDA is just still like the premier regulatory agency in the world that unlocks one of the biggest markets in the world for your pharmaceutical. But the reality still remains, right? Like AstraZeneca is coming in and dropping $2.1 billion on an asset that has gotten through clinical testing a lot faster in a different part of the world. And I think that does have some serious implications for how biotechs and therapeutic developers think about their pipeline going into the future.
Yeah. I also, I've got a lot of opinions on this that are probably, again, like wildly uninformed. It would be interesting to have somebody who is like down in the dirt with this stuff to kind of explain it to me, because my dumb like unga bunga brain is just like, you have to either have the opinion that I understand, like there's 3 opinions here. Yours is probably the most reasonable one, where it's like the FDA is mildly inefficient because there was no pressure to optimize in many little areas, and they add up to being, some significant burden.
Yeah.
Fine. But I think that there's 2 other opinions. One, it's like the FDA is just a bunch of morons who don't know what they're doing, and we should just go to China because it's easier and better and faster, and like the US has no clue what they're doing, which like I'm not sure that's true.
Yeah, agreed.
Or number 2, go and test it on like Chinese people because they're Chinese people. And if it works, great. If not, it isn't happening in America. So like whatever.
Right.
Which also not good.
Yeah.
Right. So like, I don't know.
Yeah, that's a fair point. I think, yeah, like you kind of have to walk partway down the middle. Yeah, you do have some advantage of doing it in other places of the world where you have different mechanisms that allow you to get to clinical endpoints faster, but those are going to have some trade-offs. But yeah, it's just going to be interesting to see where this shakes out, right? Because I do kind of see an analogy to what's happening with China in biotech to like the US Russia space race back in the 50s and 60s.
Sure.
Right? Where if it was just America after World War II, just kind of.
I would argue that that space race led to the US faking the moon landing. So bad.
Shut up. Oh, great.
I hear you. Competition breeds optimization.
Exactly.
So I get that there needs to be other prominent, agencies who are doing this to have that healthy relationship. But it is a balancing act.
Correct.
It's touchy when it's easy when it's like, oh, we're going to be making cars or computer technology. It's a different kind of balancing act when it comes to like human therapeutics.
Sure.
You don't want to like play fast and loose with that kind of stuff.
Yeah. Oh, for sure. Agreed. But yeah, again, right. Again, if you just kind of come back to the analogy of the space race, you see an incredible amount of investment, truly crazy innovation. Like from 1940 to like 1960 was just such a wild time of all sorts of crazy innovation happening. And then the US wins the space race in 1969. And then you kind of just see like a plateau.
Sure.
There's not the same urgency. Russia has greatly, pulled back on its space program, so there's no longer the same urgency there. And so ultimately, I do kind of see this as a good thing where competition breeds all sorts of really incredible innovation that pushes both to be a lot better.
I heard recently that like the space race, like arms race, ended up leading to advances in all kinds of other kinds of technology, not just like space.
Oh yeah, like
Associated,
we have
like,
MRIs.
computation.
We have MRIs because of the space race.
Yeah.
Waters, like the big company that does all sorts of like analytical devices, they recently launched a device called CDMS. I forget what it stands for now, and I feel awful because I helped them with some marketing for that.
Oh no.
On air. Hopefully Matthew Lauber doesn't see this. But the technology for that device was initially born out of analysis of space dust related to NASA programs in the 1950s and 60s. Yeah. So again,
wild.
Yeah, super
Yeah,
cool to see all the follow-on applications from that.
Yeah, I mean, like, I understand kind of like go to the moon, collect some dust, bring it back, and we'll see what it does. It's like in your head, you're just like, what? They're gonna find a bunch of like rocks. What's gonna help with that? But again, my dumb Unga Bunga brain is just like, it's just gonna be a bunch of rocks. But there's smart people who are like, yeah, but they're good rocks though. We need those good rocks.
Exactly. Cool. All right. So next signal here. If we're moving on very quickly, Jeff, can you bring up? I'm gonna add you to stage here.
Who to be?
It's going to be Ovid Therapeutics.
More neurology.
Yes.
For rare epilepsies and neuronal hyperexcitability disorders. Ooh.
Yeah. So Ovid in particular, they're going after rare neurological disorders. The modality is small molecules targeting the CNS. And their core bet historically has been modulating inhibitory signaling in the brain. And they had a big molecule that went through for quite a long time called gabaxadol. And it was a selective delta subunit GABA A agonist. But anyway, all that being said, they have had a rough stretch. Their gabaxadol program was an Angelman dopaminergic neurons. Still can't do it.
I was gonna say that was wrong.
But anyway, they've been reorganizing around a new pipeline of assets and partnerships, and they've had 4 different C-suite exit at once, including the CBO and the CFO. So this is just a major signal. It's like, what's going on here? Are they planning on getting ready to be acquired, and the people who want to acquire them want their own leadership in? Is this sort of change just fully needed in order to repivot into the new therapeutic areas that they're going into? It kind of remains to be seen. But yeah, they're kind of pivoting away from Angelman and the asset they had there that failed in the phase 2-3 section, and going after some of these rare neurological diseases instead. But yeah, not something you see every day where 4 different C-suite executives leave.
Yeah, it's like a new company, basically.
Yeah, exactly. Which kind of makes sense when we look again at the big pivot inside of the organization.
Yeah. Interesting. Yeah, I don't know a whole lot about rare epilepsies or neuronal hyperexcitability disorders.
Yeah.
I'm going to guess it has to do with like those kinds of things, right?
Yeah.
Well, we have some time here. Let's just like pop around and do some filtering. How about?
Sure. We have Alagos that we're going to talk about next, and they're right at the top.
Oh, okay, perfect. Well, there you go.
All right, let's do it. Perfect. Okay, so yeah, folks, just again, a reminder, in about 10 minutes or so, we're going to be having a conversation with Reagan Jarvis, the CEO of Anaka, doing some really cool stuff inside of the TCRT space. So stay around for that. But in the meantime, we're going to be focusing just for a couple minutes here on the company that has reached the top of the Biotech Voyager leaderboard for early stage biotech, Alagos. And in particular, the big signal here is that they licensed Pevivo score.
This is like, it's like reading the Old Testament. It's like every third word is like, I can't pronounce this.
Pretty much. But anyway, they've licensed it to Amoytop in Greater China for a total of $445 million in upfront and milestone payments, which just is a massive gob of money for something like this. So just in terms of where this company is coming from, like what's going on here, they're going after chronic hepatitis B. And they're using, again, an antiviral small molecule, a lot of different small molecule names today.
I'm so dumb, dude. Oh my gosh. I'm like, is this meant to be like a thing for gene therapy development?
No.
It's an antiviral, you moron.
And in particular, chronic hepatitis B.
Got it. My brain is just not working.
So there's an interesting little detail that we'll get to here a little bit later as to why there might be so much interest with this with regards to China. But overall, the really interesting way that this molecule works that they're focusing on, it's a capsid assembly modulator. So HBV replicates by packaging its genome into a protein shell called the nucleocapsid. Capsid assembly modulators like this molecule that they're going after interfere with that process. So they cause the capsid proteins to misfold or assemble incorrectly, which then disrupts the virus's ability to replicate its DNA and produce new virions. So the question becomes, Jeff, why would there be so much interest in this? Obviously, it's still a good idea, like antivirals are a good thing. But interestingly here, the reason why this might be such an interesting signal is that China carries one of the heaviest hepatitis B virus disease burdens globally, which didn't see coming. But that seems to be why there might be some interest in this. This seems to be a really big problem inside of China in particular. And so there's a larger addressable market inside of one population, inside of one regulatory jurisdiction, being China, than there might be in other parts of the world, which I did not see coming. So yeah, that's that could be one reason why there's such a such a large amount of money being put into an antiviral that has a very large disease burden, specifically inside of China.
Yeah.
So yeah, you heard it here, folks. HPV, a lot of that over in China. So be careful, I guess, if you go for a visit.
They have they got FDA fast track designation earlier this year. Blah 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 blah. Phase 2 is updated to reflect a. Okay. Okay. Yeah. I mean, again, I don't know a lot. I've got to like start looking into like other things other than cancer therapeutics and like advanced therapeutics, because these things seems interesting to me.
Yeah.
Seems pretty. You always wonder, like, how do these different antibiotics work? How do antivirals work different than antibiotics? As a kid growing up, it's kind of like, if you're sick, just take medicine.
Right.
It's like there's no difference between antibiotic or antiviral. I mean, obviously there is, right? But anyways, so yeah, I don't have a whole lot of thoughts here. They have a lot of clinical trials open right now. This view is tough to like see the actual trials happening.
That was one of the other reasons I picked this company. Like, yeah, they've just got so much going on.
And it's this one. It's this one here. Like they got the license in China, right?
Yeah. But yeah, like, that's a massive pipeline of different assets. That's almost a little like. Is that almost?
They got 4 active here.
Oh, okay. I was going to say, is this a little bit over leveraged, maybe? Just given that there's a huge emphasis right now on getting one asset through the clinic and kind of validating your platform and then going back. But if they've just been able to get a big gob of money for their lead one, I guess that kind of opens up some budget for additional pipeline considerations, right?
Yeah, I'm wondering like what the difference between these two things are. ALG. It's all based on clinicaltrials.gov, so they're registered clinical trials. It's just like this is their main one here. Like this is their phase 2. Like this is their lead asset here.
Right.
Anyways, yeah,
cool.
Pretty cool.
Well, yeah, in just a couple minutes, we will be having a conversation with Reagan Jarvis, the CEO of Anaka.
Is it Reagan or Regan? I think it's Regan.
Well,
we're going to find out soon.
Mr. Jarvis is the CEO of Anaka. It's going to be coming on the show in just a couple minutes, so stay around for that. And if you have any questions, please do feel free to drop them in the chat.
They should have a phase 1. Yep, their phase 1 started last month.
Yeah.
Cool. Anaka.
Looking forward to that. So we will be able to cover that a little bit more. But maybe just one story that we can get in the gate here before our guest comes on is Vaccinex, Jeff. They also came pretty far up the list.
Where's that? Oh, here it is.
Vaccinex. Yes. Okay, so this one's interesting. Going after Alzheimer's. In particular here, what's really interesting about this story, aside from the fact that they're going after Alzheimer's, is that this is a monoclonal antibody. So we've talked about Alzheimer's like a decent amount, I would say, recently on this show, Jeff, right? And this
a lot of neuro. Yeah, there's a lot of like neuro stuff happening right now.
Yeah. And in particular, right, one of the implicated disease pathways of Alzheimer's is my least favorite word in the whole world, dopaminergic.
You still need to go to like speech pathology for that one word.
Yeah.
Dopaminergic.
Dopaminergic.
That seems to be neurons. The hardest part there is the manure.
Yeah.
Dopaminergic.
Dopaminergic neurons. There we go.
Dopamanure.
Don't make fun of my speech impediment. But anyway, so they are developing a monoclonal antibody, and it targets the SEMA4D. So it's a signaling protein that, when overexpressed, activates the astrocytes and microglia in ways that suppress their normal neuroprotective functions and drive neuroinflammation. In Alzheimer's, neuroinflammation is a major disease accelerant. Activated glial cells damage synapses and contribute to neurodegeneration. So blocking this receptor with this antibody is meant to normalize the glial activity and preserve synaptic integrity. So again, right, there's a lot of application here with regards to just the different multifaceted components of Alzheimer's kind of all coming together and trying to come up with a solution here. We'll see what the best approach is. A lot of people coming at this from different angles.
Yeah, so one of the things that I've been seeing in neurodegenerative disorder therapeutics lately, and I've mentioned it a bunch of times, but it's worth repeating again. The pathways that are the pathological pathways involved in neurodegeneration are typically also involved in homeostatic function. It's just that when they get overstimulated, or normally it's overstimulation that causes, neurodegenerative cascades to occur. So blocking those things outright is just not a therapeutically viable option, right?
Right.
So because if, for example, with glutamate, a lot of glutamate can cause neuronal death. So you think, okay, then just block glutamate signaling. Well, the neurons need glutamate to work. So if you block glutamate signaling completely, then, they're not going to work. And normally it's not just like, oh, it's too much of the normal glutamate receptors that are activated. So just like maybe block a little bit of them, but that's not normally how it is. It's normally what happens is you get overproduction of some kind of neurotransmitter or something, and it actually activates a different kind of neuronal cascade. And figuring those things out to develop targeted therapeutics is what the enabling technology seems to be in a lot of neurodegenerative therapeutic discovery processes. So it's one of those really interesting areas where like basic preclinical research results in really tangible benefits in biotech.
Right. Yeah, that makes sense. Excellent. Well, ladies and gentlemen, Reagan Jarvis has entered the green room, so we're going to bring him on in just a second. He's the CEO of Anaka, a really cool company working in the TCRT space. They just had the initiation. Is it the initiation or the first dose, Jeff?
Dose their first patient.
Dose their first patient, even better. So yes, with no further ado, let's bring Reagan on to the show, shall we?
We shall.
Excellent. All right. Reagan, welcome to the show.
Hey guys, nice to meet you.
Thank you for coming. We appreciate your time. We'd love to learn a little bit more about what you're working on. Can you quickly introduce yourself?
Yeah, so I'm Reagan Jarvis. I'm the science founder and CEO at Anaka. Background biochemistry, cell biology. A couple of postdocs, ended up here in Sweden to start up the company in 2014. So I've been in operation quite a long time now. I was the chief scientist originally, took on the CEO role something like 8 years ago now.
Excellent, very good. So Jeff, go ahead.
Yeah. So what's your background scientifically?
So Yeah, I trained in a biochemistry department working on signal transduction pathways, specifically dithiol disulfide exchange mechanisms and signal transduction.
Boring.
Very niche, very niche stuff.
Yeah.
But, very cool stuff. I did a short postdoc in T-cell immunology, clinical immunology, and then back to signal transduction research, again, cell biology, for a second postdoc before I started at Anoka. So been wandering around, cell biology, molecular biology, fundamental biochemistry before Anoka.
So what's more interesting, glutathione signaling pathways or T-cell biology?
I was working with thiol peroxidases, not glutathione.
Whatever.
T-cell biology. I mean, if I knew how cool immunology
Hey.
was, I would have done that undergrad, to be honest.
For
I
sure.
never did any formal immunology training. But
Yeah,
it's super
I started my like academic journey doing replication stress and like helicase dysfunction in bone cancers, and then moved to macrophage biology and like dendritic cell maturation, and I was like, this is way better.
The immune system's cool, man.
It is.
Excellent. All right. So, sorry, is it Regan or Reagan?
It's Regan.
Regan.
I knew it.
I knew. I messed it up earlier. My apologies. All right. So, just with regards to like the problem that you're working on at Anoka, can you just kind of give us an overview of the disease and kind of what the opportunity is there in terms of like current treatment options and that sort of thing?
Yeah, I mean, we can get to what we're doing clinically right now and the cell therapies, but the problem we're solving as a company is much more general. So our platform's built to solve the problem of mapping T-cell targets, so HLA-presented peptides, and basically deciphering their recognition through the T-cell receptor, which is the recognition receptor of T-cells. So what we've actually done is built this massive cell biology, molecular biology platform based around highly engineered human cells, so we can compile very high precision, very standardized cell-based assays to read out target presentation, target recognition. And that is actually the fundamental barrier in any targeted T-cell therapy. It's the fundamental barrier in any vaccine, in our opinion, tolerizing therapeutic, and it spans disease areas across oncology, infectious disease, autoimmunity, allergy, transplant rejection. So that's the majority of human disease. And the fundamental barrier here is understanding the disease targets and building targeting constructs. That's what we do as a company. In terms of what we've deployed that towards in the first instance, it's TCR-based T-cell therapies. So it's like a CAR T, but instead of a synthetic chimeric antigen receptor, we use a T-cell receptor, the native targeting system of T-cells. And essentially, we've built a very, very deep pipeline on a range of tumor-selective targets. The first few we've taken off the rank are mutant KRAS in different HLA restrictions, and launched the first TCR-T trial into pancreatic cancer, pancreatic ductal adenocarcinoma. Non-viral gene editing manufacturing approach, which we've built out in-house, our own facility, our own methodologies. It's the first ever gene-edited T-cell therapy to reach clinic here in Europe. Dosed the first few patients now since the announcement a few weeks ago. And so that's looking cool. The opportunity really is to bring benefit to this very, very high unmet need indication, and that's the promise of TCR-T in general. It's very tumor selective. If you do it right, it's very safe, low side effects, and has the opportunity to be very durable and ultimately cost effective in terms of patient management.
So your discovery platform, I'll just be honest, it seems very complex. Using these dendritic cell APCs to get optimized TCR formulations and things like that, and TCR structures. Maybe I'll skip that for now. You mentioned vaccines. You mentioned that this is the primary limitation of vaccine technology. Can you guys reverse engineer your TCR constructs to get peptide like vaccine constructs as well?
It's actually simpler than that. So if you want to provoke say specific and durable immunity, you need a T-cell component to a vaccine. I think that's fairly demonstrable from historical data. So what we do as a company is map the HLA peptide targets in a first step of a cell-based assay. We build lots of T-cell receptors, and at first they go downstream through characterization and directed evolution and so on. But for a vaccine, basically we use those TCRs as the source of truth. So you validate immunogenic targets derived from a viral open reading frame, for example. And we can actually take that a step further and use those those TCRs as reporter reagents to optimize the vaccine construct as well. And that's applicable in oncology, infectious disease, and other areas as well.
That makes more sense. Okay, so it would be okay. You're not like actually, well, you would be kind of discovering vaccine formulations, but it would be like optimizing the ideal like immunogenicity of a vaccine.
Yeah, selecting the right peptide targets,
Yeah.
peptide target space from the virus, for example, and then optimizing that construct. You know,
Right.
a step further, I mean, you can use the TCR biology. We've got a huge amount of data in our custom software ecosystem. We started to train models. So if you can model this HLA peptide TCR axis more generally, you can do all that in silico, and you can pop out, hey,
Sure.
here's a sequence input, here's a nice vaccine construct. But we think there's a direct through line to that. It's non-trivial. Like you say, it's complex. It's a complex approach. It's complex biology, and it's complex coding as well around that.
Mm-hmm.
Believe it. Okay, so if we just back up for 1 second, right? For the trial that you have currently open and you've been dosing some patients, pancreatic cancer here. Why have other approaches failed in this area?
I mean, there's been some success recently. RevMed got some fairly decent data in PDAC with their pan-mutant KRAS inhibitor. I wouldn't say it's a slam dunk. It's a good, a notable PFS advantage in that phase 3 data they reported.
That was the standing ovation, Ben.
Yes.
It was a standing ovation. I think there's a lot of room to improve, right? And essentially why we're digging down into pancreatic cancer with mutant KRAS targets is because we think we can bring large impact in that setting, as many solid tumor settings, because it's basically about finding tumor selective targets, building good quality receptors, obviously, but also good quality cell therapy products, and getting them in rational trial designs where you can show that impact in a reasonable time frame, and then drive product development at reasonable cost ultimately.
Mm-hmm. So I don't want to like dig too deep into the science, but you mentioned that you guys do targeted evolution of your TCRs. Can you elaborate a little bit on that? Do other companies do that as well? Is it less efficient because they don't have a real time like reporter system to look at, or what is the advantage? Is there an advantage with your approach to allow you to do that more efficiently?
Yeah, so I mean, historically, this is our critique back in 2014 about the whole field, is that people have been treating T cell receptors like they're antibodies, right?
Mm-hmm.
Recombinant display systems, measuring affinity, and so on and so forth, and that's simply not relevant to the cell biology at the center of this. The target presentation is extremely complex cellular processes, multiple compartments, all enzymatically driven. TCR recognition is an engagement, synapse building, feedback feed-forward, rafting, all these cellular dynamics involved. So what we've done is basically, instead of using recombinant display systems, we use living cell display systems,
Mm-hmm.
and use functional challenge of the cell arrays, and basically can optimize on function rather than affinity, because that's what we want to do. You know, we're looking for sensitivity, we're looking for specificity in that cellular context. So that, plain and simple, the advantage of what we're doing. We're actually harnessing the cell biology step one, rather than having to backtrack and validate that downstream once you use recombinant systems.
Yeah, this is like one of our underlying theses here that we've kind of been hitting on a lot, is that biology is complicated. And normally the best way to test whether or not you're influencing the biology that you want to influence is to do it in the canonical setting, or as close to as canonical as possible. And I guess in the past, it's, doing an affinity assay is good enough, but if you can do the actual APC engagement and like get the biology closer, it's going to be more accurate. Do you guys know, like, where, because we had a company on recently who was doing like hinge regions and transmembrane domain mutations, and they're seeing some really interesting things. So do you guys influence where the mutagenesis occurs, or is it just like let them go crazy and see what happens?
We constrain all our mutagenesis in the CDR3 regions, the TCR.
Okay. Yeah, that's
for
fine.
two reasons, two fundamental reasons. One is, if you start tinkering outside of those regions, you drive all sorts of crazy off-target effects. And this is one of the key challenges about TCRs, is the system is inherently promiscuous. That's just how TCR biology works. So if you drive promiscuity by tinkering outside that region, you have risk of a very real risk of off-target toxicities that you can't control for. Otherwise, we don't modify outside the CDR3 region because we want these to be entirely self sequences. We're trying to basically replace TCRs as natively as possible and basically engraft those into the patient immune system.
Sounds good to me. I mean, I think I don't know of any other companies who are doing like real like I'll call it real life APC based I'll call it screening methods essentially. And like even like I go to your guys' website and it's got these crazy animations of like the things happening, and I'm like, it's actually accurate to what's like the animation that I'm watching. It's like this is kind of what's happening in their screening assays. That's pretty cool.
Yeah, we put a bit of effort into the website because we've been a long time trying to explain to people what we do. And you sort of hit it on the head, Jeff. It's a screening platform based on the cell biology. That's what we do. And, go ahead.
Well, that would be interesting to me, right? Like, so with this approach, right, when even with like TCRs versus antibodies, right? Like everyone kind of, even from early stage high school or whatnot, would understand how an antibody works. TCRs are a little bit more difficult. So kind of like, what do you see as like kind of an analogy there that works well in explaining the difference between standard antibodies and TCR-Ts here?
I've tried many different analogies over the years. I've never come up with a good one. So what I've tried to do more recently is just tackle it straight on, say what are the differences, and trying to explain that biology. And I think at the end of the day, the best explanation is antibodies stick to stuff, right? That's what they do. That's essentially their molecular function and their function in the system. They decorate targets, plain and simple. And amazing biotechnology platforms, late 90s, early 2000s, to harness that drug development, reagent development, and so on. But it's really simple because you can rely on these recombinant screening methods, basically biochemistry. The difference with a TCR is that it's not about sticking to stuff. It's a signaling receptor, right? It's engaged. Well, that T cell comes in, checks HLA peptides on the surface of a target cell, whether it's a professional APC or a stromal cell. And then once recognition is triggered, which we don't know how that happens, by the way. We don't know how a TCR signal is transduced to the inside of the cell of a recognition, which is
biophysics.
Some sort of magical mechanism. Well, what happens?
Those terms are basically interchangeable.
So you get this initial recognition, you get more TCRs flooding, this rafting, this big signalosome formation, feed forward and then feed back. And so it's just this really intricate detection system, right? And the molecular setup is quite similar to an antibody. It's VDJ recombination. It's an immunoglobulin protein, but it's inherently a different thing. It's not a binder. It's an engager and a signaling receptor. And to boot, it's not just a TCR floating on the cell surface. It's got dozens of co-receptors and other factors that are assisting that TCR to not just engage, but then reinforce that engagement.
It's how's this for an analogy? Let me know how dumb this is. It's kind of the difference between hitting somebody with a spitball or giving them a handshake.
It's not bad.
Close enough.
That kind of works.
Yeah.
Right on.
Or punching him in the face and giving him a handshake.
Nice. Okay. So just I guess with regards to the history here, just a little bit, Regan. What was the initial data that you saw with this approach that really kind of made you guys sit forward and say, "Man, we need to make a company out of this. This needs to become a clinical asset." Yeah,
I mean, this unusually is not a spin out from prior research or a spin out from an academic lab. It was born of frustration. I worked with T-cell biology for a couple of years and was like, this is pretty boring. We can't do anything specific. We can't map targets. We can't look at TCR specificity. It was all cellular profile, phenotyping, things like that. And so it was an idea on paper that we're going to build a essentially analytical engine to build the data we need to make empirical decisions. And along the way, we thought, hey, there is so much data here. Let's build software around this and make machine learning models. So I was lucky enough to meet my co-founder, Mikael Blomqvist, who's a Swedish entrepreneur, who said, that looks cool. Here's a bunch of money. Go get it done. So it's a very unusual genesis story for the company. I think along the way, like it was the first few years, we'd start building all the fundamental tooling. And there's a few results in there, particularly around the peptide mapping and validation, but also the directed evolution platform that we started developing, which was like, hey, wow, we've got this biology on a string. Let's double down and start building the products and the manufacturing around this.
That's awesome, because so one of the big things in the age of AI now is you need to be a successful company. They say you need to have good marketing or proprietary data. So people will go, well, I can make AI. I have to go now and do good marketing or get proprietary data. But it seems like you guys kind of came from the opposite way, where it's like, we have this idea, we're going to have to make this big data set. And then, oh, wait, we have the proprietary data. We can apply an AI ML model over top of it. And now you're probably entering an era where it's like, we have a really powerful proprietary data set and the system to produce more of it. And now we can utilize the advantages of the advances in AI to make use of that data even better now.
Yeah, I mean, that's dead right, Jeff. I mean, the recent advance in the LLM and literally harnessing those with different tools has seen us at a point where, hey, we've got that proprietary data set, as you say, the means to scale that data in any dimension we like and start building some very sophisticated models. And that accelerates product development on a personalized basis. We match the product to the patient with a diagnostic. We think it gets us all the way down to individualized, so patient by patient.
Yeah. Could you do hang on, how would it work? Patient, hang on, there's something boiling in my head where you could probably get to a point where you could do some kind of patient specific, like a biopsy dependent. Oh, okay, help me out, Regan. There's got to be something there.
Well, this already exists. People do biopsy dependent personalized vaccines. They're actually individualized because they're individual. We think we can get there for cell therapies as well, the TCR-T cell therapies. There's quite a lot more biology and machine learning build around that, but we think we've got a direct line to that basically AI application layer.
But I'm thinking that you guys could utilize the proprietary data set that you have to just do, like, based on tumor screening, you could use, like, independent of doing any kind of like biological assays, you could leverage your data set to maybe get something that's better than using a publicly available data set.
Oh yeah.
That's what I'm thinking. There's something
that's, yeah. The data set's not quite big to cut the lab out of the loop quite yet.
Sure.
But I think we can get there.
Okay. Interesting. So yeah, like with regards to like the results you've seen so far with the patients, how has that been going? Or if you're allowed to talk about that.
I'm not allowed to talk about that.
Okay. Everyone is cured. I can talk about it.
Yeah. Well, the screenings go well, recruitment's going well, number of patients dosed. We should be able to talk about it second half of the year.
Nice.
Right on. So yeah, like with regards to this approach, so you're starting with pancreatic cancer. Obviously, you want to validate this asset in this indication first. But like, where else would you guys want to go in the future here?
Well, the thing about TCR-T is it really spans a lot of target space. There's just a huge amount of HLA peptide target space in solid tumors, and it spans indications. Almost all indications you can name will have viable targets. So what we're trying to do is hit scale. So our first program is actually an umbrella trial where we're bringing multiple assets into the same clinical protocol. So multiple HLA peptide targets, different TCRs, all mutant KRAS in this instance. But we're looking to marry this very scalable discovery with a novel way of approaching a scalable manufacture. And I don't mean scale up of a product, I mean scale out of multiple products. So we can bring more TCRs, more TCR-T products to bear in selected indications. And why we're starting with just one indication, pancreatic ductal, is that instead of big basket trials with noisy signal and complex prior lines of therapy, complex patient management across the trial, we want to drill down to single indications with an umbrella of products and sort of span or scale out across that dimension. So we've got a number of options. We say over 40 assets in the pipeline, which seems ridiculous, but that's how this modality works. You need to drive scale across the target space and build personalized libraries and perhaps ultimately individualized TCR-T products. But we're trying to right now design trials around selected indications which make sense for the assets that we've got in our pipeline, and that's partly on the biology of the targets. It's partly on the biology of the tumors, and we're just trying to be smart about that so we can demonstrate impact in early stage trials and not spend too much money and too many years generating data. Essentially, these are very focused trials. It's a personalized therapy. It's precision therapy. You need precision trials to demonstrate your point and drive product development to commercialization.
Yeah. So there's one thing that me and Ben have been talking about recently, and this is a little bit off topic, but I'm sure that you're aware of this and the possible benefits that it could have for a company like yours. We've been looking into like the dark genome, the dark proteome, and in that same thread, just like the major advances in like antigen discovery, from being able to do single cell sequencing of tumors and AI based antigen discovery platforms. Are you guys excited about the advances happening there? Is there plans to like take advantage of that in the future? Because I feel like a company like yours is primed to be like, hey, we'll partner with these discovery platforms and like just really expand our library.
Yeah, I mean, our discovery platform does that. I mean, our thesis is how you take tumor selective gene sequence, and that parameterizes our discovery. So the dark genome, it's a cool term. The dark antigens is a cool term, but it's just antigen as far as we're concerned.
Sure.
Right. It's a non-coding sequence that's actually coding all of a sudden. It's a missense mutation. It's a splicing event or whatever it is. It's still just tumor selective gene sequence, and people. It's a good marketing hook, dark antigens, but at the end of the day, it's just antigen. It's tumor selective sequence, and we have some work in that area, but it's no different to us than say a classic RAS mutation or a MAGE or a PRAME. It's just tumor selective sequence.
Sure. So there may be less tolerance to get over. Maybe there may be a little bit easier to target those because of things like that.
Yeah, I mean, much is made about not being able to make high potency TCRs, or people often say high affinity TCRs for self antigens like PRAME and MAGE and so on. That's not our experience. And in the rare case that it is, we can break that tolerance with our directed evolution and cross-checking the safety there. So
sure,
I think there are targets that are very difficult to make potent and stringent TCRs for, but in our experience thus far, it's got very little to do with the selfness of the sequence, put it that way. Having said that, if you've got a completely non-self viral sequence or a missense dark antigen, then it's easier to make TCRs. I mean, they're not negatively selected in the thymus, away you go.
Right.
But most targets that we've worked with so far, we can make high quality TCRs with a very small handful of exceptions.
There you go. Ben?
Nifty. Okay, cool. So one question, Jarvis, that we try to ask everybody on the show.
His name is Regan Jarvis.
Did I do it again? I'm sorry.
There's the AI. There's like that AI, like, oh, talk to Jarvis. So it's just like in everyone's head.
I get it a lot. It's surprising.
Yeah.
Dear. But anyway, yeah, a question we try to ask each guest who comes on the show, and it can be a recapitulation of what we've talked about already. But the question really is, what's being slept on in biotech right now that you think is going to be a big deal in a couple of years?
Targeted T-cell therapies.
I love it. I knock on the door.
You look at the cell therapy dynamics over the last decade, right? So CAR T approval 2017, nearly 10 years ago, absolutely transformative for a small number of B-cell malignancies. And what happened since then is that hundreds of billions of dollars spent into new manufacturing tech. We can manufacture the targeted T-cell therapies in all sorts of weird and wonderful ways, but now we've got 7 or 8 approvals. It's just CD19 BCMA. We've got 1 TCR-T approval. The target space is in TCR-T, and with so much manufacturing tech sitting there ready to drive this forward, what we need is novel HLA peptide targets or tumor selective HLA peptide targets and optimal TCRs plugged into those manufacturing platforms. I think, like I said, we saw an approval a couple of years back from Adaptimmune, Amadix moving forward with a very nice PRAME product in melanoma and a couple of other indications. I think this is ready to be supercharged with all this investment in manufacturing. and very few people or companies working on what to actually parameterize those cell therapies with. We need something other than BCMA and CD19.
Yeah. So I have two very quick questions. With your technology, discovering of the TCR, the actual TCR constructs, is there potential to do things like in vivo TCRT?
Absolutely, yeah. I mean, we have some programs developing that internally. So other than the discovery platform, we've put a lot of effort into building a pipeline of manufacturing technology as well. Today, we're working with ex vivo autologous gene-edited manufacturing. We have a collaboration in the iPSC-derived allo space. We're building some new technology to solve some of the complexities around doing in vivo TCR versus in vivo CAR. And that comes down to basically TCR replacement. For a CAR, you can just sort of plop it on top of the existing TCR and away you go.
True. Yeah. Good. Okay. Yeah.
Other ways through that. But absolutely, we think that's part of the future here. Today, autologous ex vivo cell therapies are safe and demonstrably effective. That's where we are. We're looking very much forward to the new generations of manufacturing, and we're kind of agnostic to where that goes. You know, we're a discovery platform first and foremost. We've built a cell therapy manufacturing capability and technology pipeline because, hey, you need to mobilize these at scale as well. And there's a wealth of technology sitting out there that's frankly unused and underutilized because you can't build novel products around CD19 and BCMA.
You can't? Come on. Everyone else seems to think so.
Let's say it's a fairly crowded space in oncology, and
yes,
it's become an extremely crowded space in the
autoimmunity. Yeah. Yeah. Well, the other question that I had was about the personalized. Are you guys paying attention and like hopeful in the personalized vaccine space? Because if that proves to be successful, I feel like personalized TCRT is the obvious next step.
Yeah, I mean, the personalized vaccine space, or individualized, where you cook it up for every individual, is, I mean, it works. I mean, especially in the adjuvant setting where you're trying to suppress minimal residual disease or have recurrence. More data accumulating out of, I'm blanking on the company's name, European.
Moderna?
No, for disease interdiction and Lynch syndrome. So it's basically saying, hey, we're going to prevent occurrence of tumors in Lynch syndrome patients. With the advance of like ctDNA screening and all those types of things, you know, it's a very short line to disease interdiction. So we detect tumors before they become big, gnarly, malignant things, and we intervene with the vaccine. We're also developing a pathway to combine our TCRT products with individualized vaccines to build on the CD8 T cell response that we're engrafting, but importantly, build CD4 help around that. So there's all sorts of things to do here. We're very, very strong believers in individualized and even personalized vaccines in the oncology space. That same tech, that same thinking translates to infectious disease as well. So we're very active in that area preclinically, and talking to a lot of partners to mobilize those types of programs.
So I won't take too much of your time with this diatribe, but I think I've told Ben before that if there's going to be a cure for cancer, it needs to be not personalized, individualized, because everybody's cancer it's like a fingerprint, basically, right? But the problem is that with the current individualized based cancer therapeutics are vaccines, peptide-based vaccines, but peptide-based vaccines do not work the same in the oncology setting as they do in the prophylactic setting. And we need to get a really nice oomph in a individualized immune response immediately that I don't think that a peptide-based vaccine can really deliver, but a individualized TCR-T cell vaccine could.
Absolutely. I mean, a TCR T cell, you're delivering a bolus of T cells with optimized receptors against the optimal target that you've pre-selected and characterized. I mean, it's basically kicking the door down and lighting a fire in the tumor. And then you need the rest of the T cell system and the rest of the immune system to dogpile on top of that inflammation. That's how you clear tumors.
Which we learned is called chimerism recently, right, Ben?
That is
when correct. You have the patient's own immune response being kicked into gear from your T cell therapy. Yeah, they call that chimerism, apparently.
It used to be called antigen spreading as well.
Sure, that's what I thought it was called. Now it's called chimerism. We saw a press release, and it was like, oh, we have 8 out of 10 patients experiencing chimerism. And I was like, what is chimerism? So I had to look into it, and it's this new term that explains antigen spread.
New to me as well. So
well, there you go.
Excellent. Well, Regan, we want to be super respectful of your time. We really are thankful for you coming on and telling us a little bit more about what you're working on in Anaka. We will be following along in the months to come. Is there any other are there any other announcements that people should keep an eye on that'll be coming out of you folks?
Oh, nothing I can share, but watch this space.
Nice. Sounds good. That's a fair enough instruction set. Excellent. All right, Regan, thank you so much for coming on, and we look forward to following you into the future. Cheers. Thank you so much.
Thanks, guys. Good fun.
Thanks, man.
Super cool. All right, Jeff. Yeah, super interesting.
I've been following Anaka for a long time. I've talked with their CBO for a little bit about some stuff. It's really cool company. And yeah, check out. In fact, let's do this real quick. Ben, you got to see their website. The website is insane. When I first saw this, I was like, oh my goodness. You ready for this?
Yeah.
Okay. So, oh wait, hang on. We gotta find like, okay, this is okay. So you have. Let's see. This is the T cell is one of these two. I can't really tell. Probably the pink cell is the T cell. This is the cancer cell. It's going to start to die. Oh my goodness. But you've got to see their like the graphic of their in the background. You can kind of see it. Okay, here it is. Okay. So they take a bunch of different TCR sequences. They'll go and, oh gosh, I'm trying to follow this. It gives them a bunch of different kinds of TCRs. They have a whole like thing about it. But do you see how crazy this website is? Like it's
it's gorgeous.
It's super nice. They definitely have like the whole thing is like laid out in these animations. And there's one where it's like, yeah, I mean, like look at this thing. It's so nice. Anyways, I used to use this website as like an example of like what a cool website looked like to try and emulate it.
Nice. Right on.
Yeah. Anyways.
In the days of Jeff making really cool science websites,
of
which he's doing now more, if you go check out the biotechvoyager.com.
Yes, that's a pretty cool website.
Yeah, it is a pretty cool website. Do we want to talk about Tetri, Jeff, or should we leave that to another day?
Oh, definitely not right now.
No, okay.
No, definitely not.
Well, that is coming, folks. Tetri is going to be a big deal in a couple weeks / months when we launch it. It's gonna change a lot inside of the biotech space. Faux show.
Indeed.
Excellent.
Let's hope.
Well, Jeff, I know we have to wrap up a little bit earlier today. Anything else that we should focus on before we sign off?
I think it's good. We're gonna have more signals next week. I'll be back at it.
Right on. Excellent. Perfect. Well, folks, we have to drop off a little bit early today, but thank you so much for coming to the Biotech Voyager. It's the show that covers what's next in biotech, mapping the early stage signals that are going to be a big deal in a couple of years. We really appreciate your time. And if you have any recommendations of companies that should come on or people that we should talk to, do feel free to reach out to us. Probably the best place to do that is on LinkedIn. That's where we spend far too much of our time.
Ain't that the truth?
Ain't that the truth? Excellent. All right. Well, folks, thanks again for coming, and we'll see you next time. Cheers.